Rightio, folks, welcome to the last session of this study weekend, and in agreement with study weekend etiquette, we have the nicest, the coolest and the prettiest for last. So there's some laughter, no, for real. So it's a pleasure to introduce the first speaker of this session. The session is going to be about structural analysis. Our first speaker is Helen Ginn, who is a new young investigator group leader at Daisy Hamburg. And she's going to talk to us about teasing at the secrets of subtle protein dynamics. Over to you.
So thank you. Yeah, I'm your final surviving scientific co-organizer. I'm the only one left. Thank you very much all for coming and making this CSP4 study weekend the way it is. And thank you to the previous speakers for all your fantastic talks. So yes, I'm going to talk to you about protein dynamics. So I think protein dynamics we can pretty much separate into two kinds. We have the really obvious kind, like the opening of an active site that produces a large electron density change in your structure. These are really easily identifiable motions, and we deal with those pretty well. There's a second kind of motion. For example, when a ligand binds, there's a very subtle change across the entire backbone. Maybe the protein doesn't move by more than 0.1 angstroms. And so these are really subtle motions and quite hard to identify. And we don't really deal with these very well, even though they may be just as functionally relevant as the large motions. For example, the difference between a ligand binding and a ligand not binding. And generally, when you're performing research in proteins, you're going to end up collecting multiple structures of the same protein at some point, whether you wanted to or not. So when you're collecting structures, you may be able to find it easy to identify the large changes, but you may be varying other parameters as well, and these will be affecting your protein structure. Now, I'd like to talk a little bit about how we talk about these proteins computationally and how we describe them. So generally, the protein models we use are defined in terms of atom positions. Whatever model we're using, it should be good. If we want to use it to look into dynamics, then it needs to be a good model for actually describing dynamics. So for example, if we have state A and state B, the way that we parameterize the model should make it easy for us to express the change from state A to state B. And one of the questions is the atom-centric model actually good for dynamics? Now, if I am to interpolate between two states in an atomistic model, then as soon as I start to change my structure, the atoms all move too close together, and you start to violate the bond length and angle geometry restraints in your model. So I'm not a big fan of using atomistic parameterization to describe dynamics. Instead, if you define the protein in terms of bond lengths and angles, which are fixed, and then you only describe any motion in the protein in terms of torsion angle changes, then you have a distinct advantage because all of the intermediate structures have good bond length and angle geometry throughout. So that is a nice benefit. And although it comes with a lot of other fiddly problems, I do like it as a way of describing these things. And I want to talk a little bit about something that's been raised also at other conferences, which is this problem that is occurring particularly at the forefront of structural biology. And this is, you could describe it as a data to publication gap. So what tends to happen is that researchers will perform an experiment, and then you collect data from the experiment. This was a successful experiment. It couldn't have gone any better. You couldn't change anything to make it any better. And at this point, the researcher will look at the data, and then you would perform data analysis, validation, and publication. However, this doesn't seem to always happen. In fact, that data ends up going on a pile. And the researcher will go on to perform another experiment, which also successfully results in data collection from a successful experiment, and this also ends up on the pile. So there is a problem which is developing. It's particularly difficult when you're looking at subtle changes in your protein structure. And if you ask any of these researchers, why are you sitting on a mountain of data? Then the answer is usually something along the lines of, well, we can't find the narrative in the structures, we can't figure out the story to write the publication, so it'll just be a bunch of structures with no kind of theme running through the story. So we're going to do another experiment which might clarify things, but your experiment already worked, so this shouldn't be happening. And this is actually a problem with lacking the tools to really analyze your data properly. So it's my fault, it's fine. So I'm going to present the core of one of the algorithms that I have developed in order to try and mitigate this problem. So this is representation of protein entities, or ROPE, which is a clustering protocol. In here, you take all of your PDB files that you've generated that have your examples of your protein structure. From there, you can extract the very similar sequences of your proteins, and you can extract your atom positions. You can use those atom positions to optimize torsion angles with the vagabond algorithm I've developed in the past. And then you can associate the torsion angles to the sequence. We also can put in a reference sequence, and this allows us to identify mutations or deletions that you may have built into your protein structure. And this means that when we combine the two into a massive collation, then we can associate torsion angles with the residues and between the individual proteins. Now, if we linearize this table so that now every one of these columns corresponds to a residue torsion angle pair, and then you've got an entry, which is one of the boxes for each row, which is one of your protein molecules, which describes the angle for that particular protein structure. So now we have a bunch of rows which essentially code our protein conformation into the table. We can take the average of this table and subtract it, and this leaves us a little fingerprint. So this is how each protein conformation is varying with respect to the average. And we can take the rows of this table and calculate correlation coefficients, which means that we can say, are these two proteins, do they have similar conformations, or are they drastically different? We can then take those correlations and put them into a giant correlation matrix. And from there, we can do singular value decomposition. And this gives you a dot for every protein. And you can see that the similar proteins here have clustered together. So these are the proteins with similar conformations. Now, some of you may not be familiar with singular value decomposition. Sounds like maths. So I'm going to give you a little visual clue as to how to interpret singular value decomposition by describing not all of it, but the bits which are important for analyzing data in biological sciences. So what does singular value decomposition do? Let's imagine that we have observed a molecule in various configurations. This is a really simple molecule. And this uses two torsion angles to describe your structure. So let's just call those chi1 and chi2. So we've seen this in some kind of protein structure. So let's plot the graph of chi1 and chi2. And it produces this kind of graph. Now, if I ask you what is the variability of this data set, then you'll find it reasonably easy, because you can say, well, let's project all of the values of chi1 onto the x-axis and calculate the variance. Now, that's a big variance. And now we can project all of the values of, sorry, that was chi1. Now we can project all the values of chi2 onto the chi2 axis and calculate the variance, and that's a small variance. So you've described the variability in the data set. Job done, we can go home. Now, let's say that we have seen this molecule in a different binding site many, many times, and now we're going to plot another graph. And this now looks like this. So if I ask you again to calculate the variability analysis, then you will take all of the values in each of the axes and calculate a median variance for each of the chi torsion angles. But this doesn't really capture what's going on in the data set. So what singular value decomposition can do is to reorient those axes to get rid of that correlation. Now you don't have any correlation between the new axes A0 and A1. And now if you look, it's actually the same as the graph above, but we've just rotated the axes slightly. So singular, this means that we can now understand our data set variances, that we have a big variance along the new A0 axis and a small variance along the new A1 axis. We can always relate those new axes in terms of the old axes. So for example, the big variance here is, we can see that it's correlated between a positive chi1 and chi2 value. So we don't actually lose this information at any point. What singular value decomposition can give you is a list of all of these axes, which are maximizing the variation in the data set. We have the variance of each axis and the direction of each axis, and each of these axes are all perpendicular. Now, if you want to view this in the new axis position, then this is a form of principal component analysis, which is powered by singular value decomposition. Now, you may say, well, I could already see the correlation before, so what's the point of all of this? Well, this is because the example I've given you is in 2 dimensions. But if you have a protein, then the number of torsion angles that you have is going to possibly be in the thousands. So good luck visualizing that on a computer screen. What principal component analysis can do in this case is give you a way of visualizing the data that's actually interpretable to the human eye. Now that I've covered what it does, I just want to quickly cover what to watch out for. So principal components are not necessarily the functional directions in the protein structure. So for example, if you've collected a bunch of points with a boundary condition between a high condition and a low condition, singular value decomposition will give you the axis with the highest variance, which is along the horizontal. That's not the same as the functional axis, which is perpendicular to the boundary there. So that's the correct functional axis, but SVD isn't going to give you that. In a similar vein, axes with similar variances are really ill-determined. So if you do have uncorrelated variables in your data set, then the choice of axes there, because they've got similar variances, is actually quite ill-determined. It'll probably be determined by the noise in your data set, so it will be reproducible, but it won't mean anything. And then finally, it is a linear decomposition, so non-linear features can be hazards. So if your atoms are all moving in straight lines, that's great, but it's probably not chemically reasonable. So that's something to really just bear in mind when you're interpreting data. So if you have a path of structures like this, and you look at the SVD axes, you'll get 2 variances here. You don't have to define the data set this way. You could also define it as a proportion along a curve. So do bear in mind that SVD will generally report more dimensions than you may actually want to use, and don't interpret it as the number of states in your structures, because that isn't necessarily the case either. Just always bear these things in mind. Right, so back to the biology. I'm going to give an example of DNA repair by photolyase. So in healthy DNA, we can get double thymines. When you expose that to light, then they can become a thymine dimer, which is very bad, and you need to rescue that before the next cycle of cell replication so you don't get mutations. And so photolyase can come in, it can recognize the thymine dimer, and with an additional trigger of light, which makes it amenable to time-resolved studies, then it can separate the thymine dimer back and rescue the double thymine. So work by TJ Lane, who was recently working at DAISY until recently, has been looking at the photolyase using time-resolved data. This is now in a new regime because he was doing this at the XFEL, which hadn't been seen before. So when ultraviolet light triggers the reaction, I think no, it may not be ultraviolet light, then you get a trapped excited state in the picosecond regime, chemical reaction up to about 3 nanoseconds, at which point you get the release of the product. So he looked at this in a series of structures at the XFEL. And I'm very proud my postdoc is actually second author on this paper, and they looked at this and it was very obvious that there is a change in the thymine dimer, a negative density in the middle and a positive density to one side. So they were able to show the rescue of the DNA. However, I'm interested in what the rest of the protein is doing because it's quite big. So it's probably doing something as well. The active site is at the boundary between the protein and the DNA. But what is the rest of the protein doing? So one way we could answer this is to overlay all of the structures and have a look to see if we see a meaningful, some kind of meaningful change in the data. So have a look at this carefully and try to see what the trend is between all of these time points. And if you're anything like me, you might be thinking, I can't really see any kind of trend here. And that's because it's quite difficult to see a trend in this data set. So you may conclude that there is actually no meaningful dynamics in the structure. So let's take all of those structures and instead we'll pass them through rope. And we're looking now at the rope space, which is the PCA of the torsion angles like I just described. This, let me just talk through this plot a little bit. So each of these points is equal to one of the proteins that you've observed in crystallography. And we have chain A and chain B because there are 2 chains in the asymmetric unit. We have our dark structures here. We have the 3 picosecond at the top and 100 microseconds at the bottom. And as you can see, there is actually a trend here. It's actually a little bit bigger in the chain B, even though chain A was used in the paper to show the active site region. The functional axis is on the near vertical, but there's actually some substantial off-axis jitter, and that's probably what's making it difficult to actually see this effect when we overlay the structures because there's a bit of noise. So that makes it actually very difficult to interpret by eye when you're looking at overlays of protein structures. The natural variation of the protein conformation makes it quite hard to distinguish these features. But we can make them very clear because, like I said before, those axes can be expressed as a combination of the original axes, the torsion angles, and we can view that, or we can view the atom motion associated with that. And as you can see in the top left of this video, we have a rotation of a helix, and also throughout the rest of the protein, there are hinge points where the protein is moving more than others and actually controlling the motions which are happening in the rest of the protein structure. So this gives you a handle on what the rest of the protein is doing and where you may want to look in particular at the effect of mutations at those hinge points in particular. So I also want to go back to throw back to one of the ideas I put at the very beginning of the talk, which is what if we had 2 different states of our protein and we'd like to actually develop a pathway that moves from state A to state B. So this I'm going to illustrate with KRAS because it has a very big conformational change. And my first naive thought was, well, I have a torsion angle vector that corresponds to the conformation of state A, and I also have one for state B. So why don't we just linearly interpolate between state A and state B? No, that's not a good idea. I'll show you why. This is what happens to the protein. It performs a pirouette and does very, very silly things and violates all the laws of chemistry. and physics. So this isn't actually the way to go, but at least it starts and ends in the right place. So let's have a look at the stuff in between. So this actually decomposes into two problems. So first problem is the direction of bond rotation. So if we have solved a structure in state A and state B, for any given bond, it may go from 0 to 180 degrees, but it could also go from 0 to minus 180 degrees. So which one do you choose? You don't actually see a difference. That choice is important, and so we can choose the correct one. And now the protein is definitely moving a lot less. There are no pirouettes, but it is bumping into itself quite substantially. So we're not quite there yet, but you can choose the correct bond direction to minimize that amount of motion. The second problem is compensatory movements. So although most of the loop motion is actually happening in the center of the loop, elsewhere in the protein structure, you have nonlinear compensatory movements adjusting for the fact that those torsion angles are moving. And so this allows the rest of the protein to stay reasonably still. So we can use this to inspire a different interpolation for the torsion angles, where now, instead of having a linear progression, we have a polynomial progression. And so we can adjust all of the torsion angles to reduce the amount of motion in the protein structure. How we do that exactly is that we can take for every atom, we can calculate the pathway if you take a linear interpolation between A and B. So that's just an atom moving in a straight line between the two. We also have a trajectory, as I've been showing you in the videos, which we calculate using the interpolation using the torsion angles. And if we have a look at those two paths for every atom, then the difference between those, the RMSD between the two paths, acts as a way, if we minimize that, then we're minimizing the amount of momentum in the protein, because generally the momentum will be conserved as much as possible. So this reduces the amount of motion happening in the protein. So we can minimize that. And now the protein is moving far less. So this really has minimized. There's one clash between side chains, but otherwise it's starting to produce a reasonable hypothesis using perfect bond length and angle geometry in between for how a protein may be getting from state A to state B. So just going to use this as a proof of principle that we can actually use this for refinement in electron density. Now, I don't expect anyone to actually do this in practice because who knows what state A and state B are, and there are probably not just state A and state B, but up to Z. So this is more just showing you how this kind of refinement would work. But what we could do is we could define a coordinate, which is a mean and standard deviation between state A and state B. And from this, we can sample an ensemble of protein conformations, and then we can fit the mean and standard deviation to the electron density. So this is just two parameters, and if we now have a look at a little video of this happening, this is in real time. The protein here, I've chosen a protein with state A and state B, which are reasonably similar, and you can see that it's expanding to fill the electron density using these states in order to try and fill in the bits which are more blurry and keep the parts together which are more sharp. So I don't expect anyone to use that in its current form, but this idea of creating coordinate axes where we can move a loop or switch a little bit on the backbone means that that'll open opportunities for making ensemble refinements which are totally deterministic and easy to analyze. So if you want to use rope, both of those features are now available. If you go to this web address here, if you are on a Mac, everything's fine, you have a Mac executable. If you're on Linux, there is a Linux Flatpak distribution. If you're on Windows, you can use the web app. Anyone can use the web app. Firefox works well. It's just bear in mind, it's running on your local computer. It's in WebAssembly. It only has one thread and 2 GB limit. So it's a bit of Some can be a little bit of a toy, but it's nice to get to use it. There's a link to the paper with a written tutorial. There's also a YouTube tutorial, if you prefer that format. And I will give a Zoom tutorial on the 17th of January at 1 o'clock. So put your, I'm going to ask you to send your email address in Slack for me, and then I'll send you the Zoom link, not straight away, but when I've collated all of the information together. And I'd like to acknowledge my group, my funding bodies, past and present, my group, again, and my collaborators who are my environment. Thank you very much, and I'm happy to take your questions.
Thank you, Helen, for the great talk. Fascinating. Are there any questions on the floor here? If not, then I can kick off one. Oh no, there is one. Let's go for it.
Hello. I'm Daniel Salis. I guess I work at TCP4 at STFC. I got two questions. When you do the mean subtraction, do you account for like outliers, or is it just like the straight normal mean? And for the clusters, how do you decide how many clusters to, I guess, define?
So for the first question, I will, by the time I've answered my first one, I'll forget the second one. So do bear that in mind. So actually, I'm not doing a mean of the angles, but I'm doing a mean of the cosine and sine, or even the cosine and the sine over the cosine, which means that it's actually following a two-lobed shape. So outliers tend to just disappear into noise because there's no ability to go very far away from the origin. I have forgotten the second question.
When you showed the clusters, how do you decide whether you have like 3, 5, or n clusters?
Right. So that decision isn't made by the software. So it's replotting all of the values in PCA, but there's no kind of k-means attempt to actually choose the values. So you can either do that by eye, or you can also color by metadata or change the icon and so on. But it doesn't try to identify clusters for you.
Thank you. We have another question here from Ashwin.
Thank you, Ashwin Chari, Max Planck Institute. Helen, there are many recent reports that mutations far from the active site actually enhance enzyme evolution. I was wondering, in the particular cases you've looked at, whether you have seen a correlation between the hinges you find and actually those mutations which elicit gains in enzyme catalysis?
I would absolutely love to do this analysis. I haven't done this yet, I'm afraid, but it's on my list. I mean, I have been working the other way in engineering in mutations, which have then altered the enzyme catalytic activity. So I've been approaching this from the reverse direction, but not the forward direction, if that makes sense.
Oliver, please.
So Oliver, again, as Oliver Smart, would it be of any interest when you're interpolating torsion angle space to do it, to do the interpolation using a function to make sure things don't bump into one another?
I've been thinking about ways to do this, and yes, there are a few ways, but go on, sorry.
So it didn't ever go anywhere, but I used to work on something of looking at looking at energy space and getting a transition state and going for the low energy states. There's a method which I came up with called path energy minimization, which I don't think anyone cited. It was published in 1993. If you're interested, I can't send you a reprint because I think they all disappeared at one time. But there's a thing published. I'll send you send you something about it.
Yes, please.
Yeah, it's and it was really slow at the time, but I think it would be quite fast now.
Okay, yeah, no, I'd love to have the paper or what you can find.
Let's thank Helen again and move on in for the sake of time. Our